Hello and welcome dear students. Today we are going to discuss about the topic DNA sequence variation and human origins. The main objectives of today's lecture are to know the basics of DNA sequencing, to discuss about the diversity of humans, to discuss about the distribution of genetic diversity within and between populations, to discuss about the patterns of genetic diversity and human evolutionary history, to discuss about the African origin of modern humans, to discuss about the origins and affinities of specific human populations, and finally to know the applications of DNA sequencing. Dear students, let's start with the introduction. The past 2 decades have witnessed an explosion of human genetic data. Innumerable DNA sequences and genotypes have been generated and that have led to significant biomedical advances. In addition, these data have greatly increased our understanding of patterns of genetic diversity among individuals and populations. Genetic variation can contribute to an understanding of human similarities and differences, origins, and evolutionary history. Patterns of genetic diversity provide information about population history because each major demographic event leaves an imprint on a population's collective genomic diversity. A reduction in population size reduces genetic diversity, and an increase in population size eventually increases diversity. The exchange of migrants between populations inevitably results in greater genetic similarity, while isolation preserves genetic uniqueness. These demographic signatures are passed from generation to generation such that the genomes of modern individuals reflect their demographic history. Thus, it is reasonable to say that human history is written in the DNA. As we accumulate more and more data on DNA variation and as we develop better tools to analyze these data, our history becomes increasingly clear. DNA sequencing is the process of determining the nucleic acid sequence, the order of nucleotides in DNA. It includes any method or technology that's used to determine the order of the 4 bases adenine, guanine, cytosine, and thymine. The advent of rapid DNA sequencing method has greatly accelerated biological and medical research and discovery. Following the development of the fluorescence-based sequencing methods with a DNA sequencer, DNA sequencing has become easier and order of magnitude faster. Dear students, let's now discuss about DNA sequencing. DNA sequencing refers to the general laboratory technique for determining the exact sequence of nucleotides or bases in a DNA molecule. The sequence of the bases, often referred to by the first letters of their chemical names like adenine, thymine, cytosine, and guanine, abbreviated as A, T, C, and G, encodes the biological information that cells use to develop and operate. Establishing the sequence of DNA is the key to understanding the function of genes and other parts of the genome. There are now several different methods available for DNA sequencing, each with its own characteristics, and the development of additional methods represents an active area of genomics research. Knowledge of DNA sequences has become indispensable for basic biological research, DNA genographic projects, and in numerous applied fields such as medical diagnosis, biotechnology, forensic biology, virology, and biological systematics. Comparing healthy and mutated DNA sequences can diagnose different diseases including various cancers, characterize antibody repertoire, and can be used to guide patient treatments. Having a quick way to sequence DNA allows for faster and more individualized medical care to be administered and for more organisms to be identified and cataloged. That's why we are talking about personalized medicine or precision medicine. The rapid speed of sequencing attained with modern DNA sequencing technology has been instrumental in the sequencing of complete DNA sequences or genomes of numerous types and species of life, including the human genome and other DNA sequences of many animal, plant, and microbial species. The first DNA sequences were obtained in the early 1970s by academic researchers using laborious methods based on two-dimensional chromatography. Darwin could compare only the anatomy and embryos of living things. Today it is possible to compare their DNA. Similar DNA sequences are the strongest evidence for evolution from a common ancestor. Dear students, let's now discuss about the diversity of humans. Perhaps the most widely cited statistic about human genetic diversity is that any two humans differ on the average at about 1 in 1000 DNA base pairs, that's about 0.1%. Human genetic diversity is substantially lower than that of many other species, including our nearest evolutionary relatives, the chimpanzees. Genetic diversity is a function of a population's age, that is the amount of time during which mutations accumulate to generate diversity and its size. Our genetic homogeneity implies that anatomically modern humans arose relatively recently, perhaps 200,000 years ago, and that our population size was quite small at one time, perhaps 10,000 breeding individuals. To put the 0.1% genetic diversity estimate into perspective, it is useful to remember that humans have approximately 3 billion base pairs in a haploid cell. Thus, any pair of humans differs by approximately 3 million base pairs in total. These differences contain much useful information about the evolutionary history of our species. In addition, the small proportion of differences that occur within genes can lead to critical inferences about the effects of natural selection. Dear students, let's now discuss about distribution of genetic diversity within and between populations. Human populations can be defined along geographic, political, linguistic, religious, or ethnic boundaries. Using a common definition that groups populations into major continents, Africa, Asia, Europe, and North and South America, many studies have shown that approximately 90% of genetic variation can be found within these populations, and only about 10% of genetic variation separates the populations. Thus, the great majority of genetic differences can be found between individuals from any one of the major continents, and on average, only a small proportion of additional differences will be found between individuals from two different continents. Furthermore, because human history is a history of population movement, and because humans are extraordinarily adept at sharing their DNA, the genetic boundaries between populations are typically indistinct. For any given DNA sequence or gene, two individuals from different populations are sometimes more similar to one another than are two individuals from the same population. The fact that humans are relatively homogeneous at the DNA level, combined with the fact that between population variation is modest, has significant social implications. Importantly, these patterns imply that the DNA differences between individuals and between populations are relatively scant and do not provide a biological basis for any form of discrimination. Dear students, let's now discuss about the patterns of genetic diversity and human evolutionary history. Studies of our evolutionary history have benefited enormously from analysis of mitochondrial DNA and the non-recombining portion of the human Y chromosome. Because these regions are not shuffled by recombination, they are transmitted intact from one generation to the next generation, revealing the paternal and maternal lineage of a population. Populations share mitochondrial DNA or Y chromosome lineage as a result of common origins or gene flow. Comparisons of Y chromosome and mitochondrial DNA variation in a number of populations have indicated greater levels of mitochondrial DNA lineage sharing among populations, suggesting that females may have experienced more mobility during much of our history than have males. This hypothesis needs further testing to determine how general the pattern may be. One must also exercise caution in interpreting analysis of Y chromosome and mitochondrial DNA variation, because each system is essentially one highly variable looks. Statistical estimates of parameters, for example, the age of the most recent common ancestor of a group of individuals, are highly inexact. More reliable estimates of such parameters can be obtained by analyzing multiple independent autosomal loci, that is single nucleotide polymorphisms or short tandem repeat polymorphisms. In addition, genetic data must be combined with archaeological, linguistic and historical data to achieve an accurate interpretation of human evolutionary history. Dear students, let us now discuss the African origin of modern humans. The most basic question about human history concerns the origin of anatomically modern humans. For some time, discussion of this question has been framed by two competing hypotheses. The first, usually termed the multi-regional hypothesis, states that modern humans evolved from more archaic forms such as Neanderthals in several different parts of the old world, that is Africa, Asia and Europe. The genetic similarities seen in contemporary populations are the result of gene flow between these populations as well as natural selection for similar adaptive traits. In contrast, the out of Africa hypothesis states that modern humans originated first in Africa and then migrated to Europe and Asia, less than 100,000 years ago, to replacing existing archaic human populations. Most genetic evidence supports the out of Africa hypothesis or a close variant of it. Nearly all genetic studies indicate greater diversity in African populations, which is consistent with an African origin of modern humans. Because diversity is a function of both time and population size, however, this pattern could merely be the result of a larger historical population size in Africa. The fact that most genetic variation in non-African populations is a subset of the variation found in African populations lends further support to the African origin hypothesis and is difficult to reconcile with the multi-regional hypothesis. Non-African populations also tend to be more similar to one another than they are to African populations, a finding that lends additional support to the hypothesis that a subset of the African population left the continent to colonize the rest of the world. Genetic evidence also indicates that early human population was quite small for some time, approximately 10,000 or so breeding individuals. That is inconsistent with the multi-regional hypothesis because continuous gene flow among human populations scattered broadly over the old world would require a much larger population. Although genetic data offer broad support to the hypothesis that modern humans first evolved in Africa, it is more difficult to establish that archaic populations from other parts of the world made no genetic contribution to today's populations. That is, that there was no mixing between anatomically modern humans from Africa and archaic populations from Europe or Asia. Ancient nuclear sequences are sometimes observed in non-African populations. For example, beta globin sequences in modern Asian populations that derive from a common ancestor who lived 800,000 years ago. These sequences may have descended from archaic humans living in Asia long before the emergence of modern humans. However, it is also possible that such sequences did originate in Africa and were carried to Asia or Europe by modern humans themselves. This notion should become clearer as more and more autosomal sequences are analyzed and compared in multiple human populations. Dear students, let's now discuss about the origins and affinities of specific human populations. In addition to addressing broad questions about the origins of human species, genetic data have been used extensively to study the origins and affinities of specific population groups. For example, analysis of genetic variation in Ashkenazi Jewish population show that these populations are most similar to one another and to other populations from the Mid-East and are distinct from non-Jewish European populations, although they have received some genetic contributions from these populations. Intriguingly, approximately 2/3 of Jewish males with the last name Cohen, the name associated with the Jewish priesthood, have essentially the same Y chromosome. Analysis of Y chromosome, mitochondrial DNA, and autosomal DNA support the relatively recent derivation of Native American populations from populations that lived in Northeast Asia. These data also indicate that there may have been substantial bottlenecks as the founding populations entered the Americas some 200,000 years ago. Another even more recent event in the human settlement of the Polynesian South Pacific islands estimated to have taken place just 3,000 years ago. Most evidence supports a Southeast Asian origin of Polynesian populations with mixture between these individuals and Melanesian populations as they spread through the South Pacific. An analysis of mitochondrial DNA, Y chromosome, and autosomal variation in South Indian caste population indicated less differentiation among castes for mitochondrial DNA than for Y chromosome DNA. This reflects greater levels of female mitochondrial DNA gene flow among castes than male Y chromosome gene flow. Incidentally, comparisons of genetic variation in populations from India with Roma, that is Gypsy population, supports an Indian derivation of many of these populations, which is consistent with the Roma's own accounts of their origins. Dear students, let's now discuss the applications of DNA sequencing. DNA sequencing provides the means to know how nucleotide bases are arranged in a piece of DNA. Several methods have been developed for this process. These have 4 key steps. In the first instance, DNA is removed from the cell. This can be done either mechanically or chemically. The second phase involves breaking up the DNA and inserting its pieces into vectors, cells that indefinitely self-replicate for cloning. In the third phase, the DNA clones are placed with a dye-labeled primer, a short stretch of DNA that promotes replication, into a thermal cycler, a machine which automatically raises and lowers the temperature to catalyze replication. The final phase consists of electrophoresis, whereby the DNA segments are placed in a gel and subjected to an electric current which moves them. Originally, the gel was placed on a slab, but today it is inserted into a very thin glass tube known as capillary. When subjected to an electric current, the smaller nucleotides in the DNA move faster than the larger ones. Electrophoresis thus helps sort the DNA fragments by their size. The different nucleotide bases in the DNA fragments are identified by their dyes, which are activated when they pass through a laser beam. All the information is fed into a computer and the DNA sequence is displayed on a screen for analysis. The method developed by Sanger was pivotal to the International Human Genome Project. This project provided the first complete human DNA sequence in 2003, as it was started in 1990s. Data from the project provided the first means to map out the genetic mutations that underlie specific genetic diseases. It also opened up a path to more personalized medicine or precision medicine, enabling scientists to examine the extent to which a patient's response to a drug is determined by their genetic profile. The genetic profile of a patient's tumor, for example, can now be used to work out what is the most effective treatment for an individual. It's also hoped that in future that knowing the sequence of a person's genome will help work out a person's predisposition to certain diseases such as heart diseases, cancer and type 2 diabetes, which could pave the way to better preventive care. Data from the Human Genome Project has also helped fuel the development of gene therapy, a type of treatment designed to replace defective genes in certain genetic disorders. In addition, it has provided a means to design drugs that can target specific genes that cause disease. Beyond medicine, DNA sequencing is now used to genetic testing for paternity and other family relationships. It also helps identify crime suspects and victims involved in catastrophes. The technique is also vital to detecting bacteria and other microorganisms or organisms that may pollute air, water, soil and food. In addition, the method is important to the study of the evolutionary history of different population groups and their migratory patterns, as well as determining pedigree analysis. DNA sequencing variation analysis also underpins pharmacogenomics. This is a relatively new field which is leading the way to more personalized medicine. Pharmacogenomics looks at how a person's individual genome variations affect their response to a drug. Such data is being used to determine which drug gives the best outcome in particular patients. Over 140 drugs approved by the FDA now include pharmacogenomics information in their labeling. Such labeling is not only important in terms of matching patients to their most appropriate drug, but also for working out what their drug dose should be and their level of risk in terms of adverse events. Individual genetic profiling is already being used routinely to prescribe therapies for patients with HIV, breast cancer, lymphoblastic leukemia and colon cancer, and in the future will be used to tailor treatments for cardiovascular diseases, cancer, asthma, Alzheimer's disease and depression. Drug developers are also using pharmacogenomics data to design drugs which can be targeted at subgroups of patients with specific genetic profiles. Aligned DNA sequences form the base of many analyses are used to infer evolutionary patterns and processes. Within species, this information can be used to quantify the degree of population differentiation, migration rates among populations, and even the demographic history of populations. Dear students, let's now conclude the lecture. DNA sequencing is a technique used to determine the nucleotide sequence of DNA or deoxyribonucleic acid. The nucleotide sequence is the most fundamental level of knowledge of a gene or genome. It is the blueprint that contains the instructions for building an organism, and no understanding of genetic function or evolution could be complete without obtaining this information. Since DNA is an informative macromolecule or a polymer in terms of transmission from one generation to another, DNA sequencing is used in evolutionary biology to study how different organisms are related and how they evolved. The studies of genetic variation can inform us about our origins and history. Humans are relatively homogeneous genetically, reflecting a recent common origin. Anatomically, modern humans probably originated in Africa, undergoing population bottlenecks as some left to colonize the rest of the world. Because these events are fairly recent and because of extensive gene flow within major human populations, most genetic variation can be found within major human populations. Genetic data also provide useful information about the origins and histories of individual human populations. Dear students, it was all about today's lecture regarding DNA sequence variation and human origins. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.